Here are some laboratory tests, and you have to determine the number of CD4 plus cells in the blood of a patient infected with HIV. First of all, let's have a look how the CD4 positive cell is related with the infection of HIV and why it is important to determine the number of CD4 plus cells in the case of the infection with HIV. So, that CD4 plus cell means the Th cells. That is the T helper cells. So, the T helper cells are having CD4 plus receptor. That is a kind of glycoprotein. It is a type of glycoprotein and that is present on the surface of the T helper cells. That is the CD4. That CD4 receptor is on their surface. And now, if the HIV comes, it is having another glycoprotein on the surface that is the GP120 glycoprotein 120, and its receptor is the TH plus TH cells that is having the CD4 plus receptor. So the GP120 comes and bind with the CD4 receptor and thus it internalize inside the cell, and thereafter it hijacks the cellular machinery, it replicates itself, and thereafter it will break and burst the cell. Now thus the HIV cells goes inside the TH cells via the CD4 receptor, and now thereafter the successful replication they destroyed the cells, and for the successful infection case of the HIV the number of cells will go down. So if you are having a sense on how much cells are there in a body which is CD4 plus, then you will get a look of the infection. If the infection is minute or severe, this kind of sense you can get from the count of CD4 plus cells. So to count the number of cells that is having a specific receptor, the flow cytometry is the major way to do this. So the flow cytometry is the answer. How it works? Like in the flow cytometry case, like you want to determine a specific cell that is having the CD4 receptor, and you will design a fluorescently active molecule against the receptor. Like as an example, the GP120 protein that can come and bind with this CD4 receptor. Like that way, you will make another receptor, another protein that can come and bind with the CD4 plus receptor, and which will having a fluorescent tag. Thus, if you run those culture of cells or those cells through this flow cytometer, then you will get here the laser will be there, and depending on the only those cells that is having the CD4 plus marker or CD4 plus receptor, so they will emit this light, the fluorescence emitted from stained cell, that will be detected if they are positive with the fluorescence light. So you will get the fluorescence tag from those CD4 plus cells, and thus you can count the number for how many cells are there which is emitting the fluorescent light. Thus you can determine the number. Here are certain other procedures like the agglutination, complement fixation, and ELISpot. Those all are dependent on the antigen and antibody reaction. Like in the agglutination case, like if those are the antigens and the antibodies will come like this, and they will form a precipitate. And thus from the visible agglutination of the antigen antibody, there will be a spot on the well plate. Now from the agglutination you can easily see the agglutination. That is the agglutination process. There is no relation with a specific marker or a specific kind of that receptor having cell. They only can determine whether there is a interaction between the antigen and antibody, and it should be in a sufficiently large amount. The antigen or antibody both should be in sufficiently large amount to make a visible agglutination. And the complement fixation also depends on the reaction between the antigen and antibody. So the complement will be fixed if only antigen and antibody both are present in the cell. So if that is happening, so that is the antigen and antibody reaction. If they are making a complex of antigen and antibody, thereafter we give the complement in that fluid, that oil. So there only the complement can go and bind with it. Thus the complement fixation happens. And ELISpot is totally different. It determines the secretion of a single cell, like in the well plate. So you are analyzing the what kind of proteins a cell is secreting. Like it could be antibody or some growth factors, cytokines. If they are secreting, their respective antibodies will be here, and they will come and bind here. And depending on some detection antibody, we will determine if there is your target secretion, if the cell is secreting your target molecule or not. Thus the ELISpot works. So here we got a clear vision on the flow cytometry, and we know if there is a specific kind of like CT4 or any other kind of tags like the markers to sort this kind of cells or to count the number of this kind of cells. Those are having a specific markers. Flow cytometry is the best way. Let's have a look on this technology that is based on the SELEX, that stands for the systematic evolution of ligands by exponential enrichment. So this technology is useful to identify aptamers. So what are aptamers? Aptamers is a specific kind of sequence, like it should be a single stranded DNA or RNA sequence that can identify, that means it can bind with a specific ligand. It could be a protein, it could be some chemicals, it could be the cell membrane receptors, other biomolecules like the proteins, and also the DNA and RNA sequences could be its target. So it is like the antibody which can bind with its specific antigen. So that is, so here you are designing the DNA or RNA in such a way, like that should be in a single stranded form. You are designing it in a such a way so that it can go and bind with its target molecule, target biomolecule. That is like the antibody. So what is the advantage over the antibody for this aptamer? These aptamers are very short in size. Antibodies are very big. It can show some immunogenic activities, but in respect for that DNA or RNA sequences, it can't be that much long. It should be within 60 to 100 nucleotide long. That is very much short length. So the usefulness of this aptamer relative to this antibody is very much high, and also the designing and the production cost both are less in the case of aptamer. So here, how the SELEX technology works, we have to have a look on this. So here, that is the primary pool of single stranded DNA or RNA library. So that is the pool of unknown sequences. You don't have the knowledge of the sequencer, what is present there, but that should be large in number. The pool should be large, and the pool, the library should be designed in such a way so in both the end you should have a known sequence, and in the middle the unknown sequence will be there. So why the construction should be like that? Because you when you will select some sequences from the library, from the pool, you have to identify which sequences are there. So there you have to go for the PCR. If you don't know the flanking sequences, the end sequences, then you would not be able to make the primer, and you will never be able to do the PCR, then the sequence will remain unknown for you. So here you have to identify the sequence from the pool. So what you will do, that it should be in a folding cases, because you are making single stranded DNA or RNA molecules, so that they can form the folding. They are in a single stranded form. So if there is a sequence complementary regions, so they can fold. And now that is the incubation period. So in the incubation period, you will provide your target molecule, like the protein or the lipids, whatever the molecule you want to see, you want to check, your target molecule should be there in the incubation phase. So you will incubate your primary single stranded DNA or RNA sequences with your target. And next, the unbound oligonucleotides should be washed away, and only the bounded nucleotides should be there in the pore. So now the PCR amplification will happen, that I told you earlier. The PCR amplification will happen, and you will get a sequences, and thereafter, and this, now you know the sequence, you have run the PCR, and you will run these procedures for at least 10 to 20 times. Why? Because if you are running this for that much of time, then you are actually exponentially enriching. So in the beginning, the sequences might be you might get some sequences that are not exactly complementary or exactly having the ligand activity of your target molecule. But if you are running these procedures for 10 to 20 times, then that primary single-stranded DNA or RNA sequence, that should going to be more specific and more specific to your target sequence, to get a exact aptamer, to make the aptamer too much, that to make it at least the exact complementary to your sequence. If you want to get the highest ligand intensity, like if you want to see how the specific, to make it more specific, you should run 10 to 20 times. Now you will get a enriched pool. Now you will get a enriched pool of your single stranded DNA or RNA molecule, that is known as the aptamer. The specificity is too high when you are running for long period of time. Now you will get a enriched library, and thereafter on the last round you will get, again you will sequence the target, that single stranded DNA and RNA library, and you will get the aptamer candidate, and thereafter depending on the concentration you can get which one is more there, and depending on that you can decide which one you will be preferred as the aptamer. There will be more sequences, like some should be more specific to your target molecule, and some should have less efficiency to your target molecule. So from this concentration pattern, you can identify which one is having the more efficiency towards your target molecule. So thus you can determine the aptamer. And no one that miRNA, shRNA, primer, nothing is related with the aptamer. So the aptamer is the answer. Among the following techniques doesn't depend on nucleic acid hybridization. From the name itself, you can guess that is the immunoprecipitation. That means immunoprecipitation means antigen and antibody might be involved in that case. So the chromatin immunoprecipitation is the answer. So how it works? See in the diagram. So here the cross-link between the proteins to DNA, that is the major determining factor of this technology. So here what you want to determine, you want to determine a specific DNA sequence, that is the sequence, and you want to know this sequence. So here you can do the chromatin immunoprecipitation technology. So how it works? Like if you target DNA from this technique, you can determine if your target DNA is in a heterochromatin or euchromatin position. So like the epigenetic modification, to know the epigenetic modification, you can use this technology. How it works? Like certain DNA binding proteins are there. They are having a specific kind of motifs so that they can go and bind to your target DNA. Here, you will design a protein which can go and bind to your target DNA. That is here, this one is coming here and binding with your target, this protein. And there is a like the situations are there whether it can bind with your target sequence or not. If the chromatin is in a very much condensed situation, then the protein, the your sequence is in a very much condensed situation. So that is in a heterochromatin position. So your protein will not be able to get, will not be able to access the site. So it will remain unbound. But if it is in a euchromatin condition, then the position would be open for the protein to come and bind. So here, if the cross-linking is happening, that means the that DNA is in a euchromatin condition. So this all kind of things are regulated via the histone proteins, the modification of the histone proteins. So here your protein is there and it got its site to bind with your target sequence. Now in the second option, we have to remove the remaining sequences. There is no need of these sequences. How will you remove? You can take a, you can shear the DNA or you can use some enzymes like the MNase. So MNase is a endonuclease. So if you are, so that region, your target sequence is protected via the protein that you gave to it. And this sequence will not be cut down because it is protected. But other regions, so that is not protected, that is not bounded via the via your protein, that will be degraded down, and this sequence will be there, that will cut away, that will be cut away, or it can be degraded. So now you will be get a free complex of your target DNA and protein, which will be detected via the target antibody. So you will design an antibody against your target protein. The protein should be make a complex with the DNA, and so the protein DNA complex now would be determined via the antibody. Now comes the fourth option. So here you have to reverse cross-link. So you will reverse cross-link the that that binding between the protein and the DNA, and you have to degrade the protein and the antibody complex, means the antigen antibody complex, you have to degrade it. So you can use some protease enzymes, and now in your solution only your target DNA molecule is present. Thereafter you will attach P1 and P2 adapters. Those adapters are the known sequence of DNA. So you will attach on the both the side of your target DNA, you will attach the known sequence so that you can design the primer against it, and you can run the PCR thereafter, followed by you can make the sequencing. So that is the procedure of the chromatin immunoprecipitation. Now if we look on the other procedures like the DNA microarray, so on a plate, on a immobilized plate, you will get a bunch of probes. So bunch of probes will be there. So if you run the DNA, your DNA solution, if you run over them at a single time period, you will be able to see the protein, the DNAs, those are getting bounded with the probes. So at a single time, you will be able to determine lot of DNA, which is going and binding with the probes. So for DNA microarray, you will use when you want to know which kind of DNA you are having. You are not having any kind of target DNA sequence. So you will be able to determine which kind of DNA is present in your solution. Also, you can get your target DNA sequence, but it will tell which kind of DNA against the probe. It will tell which kind of DNA are present there. From that pool, you can determine your target DNA sequence. And in the Haugen blotting, there you will use a specific probe to your target DNA sequence. So after running the agarose gel, you will thereafter you will give the DNA probe on it. Your target probe will be given after the separation of the DNA. So on the plate, your DNA was separated, and now you will give the probe, and the probe will go and bind to the specific region, and now you know your DNA is present on this sequence, on this specific region. That is the southern protein. And the chromosome painting is another technology. So against the chromosome, like if that is the chromosome, so the probe will be designed against the specific region of the chromosome. Now, from a bunch of chromosome, if you give the probe, so you will be able to determine where your chromosome is present. And it has another importance, like you know in a bunch your target chromosome is present. So if you use this probe and you are not getting the signal, you are not that probe should be fluorescently tagged to. So in the chromosome painting, the probes are generally fluorescently tagged, so that at it is saying about the painting means different kind of probes with the fluorescently tag you can use against the chromosome, and you will get the light, you will get the color, that is the painting. So in an organism, so different kind of chromosomes are there, different chromosomes are there. So if you design the probes against each chromosome, then you will get the painting, a painting pattern you will be getting. And now if you are not getting a specific signal that should be there, that means the chromosome might have gone through some chromosomal abnormalities or aberrations. So might here be some mutation. That's why this probe is not able to come and attach with this region. So that is the utility of chromosome painting technology. So this question is saying about the cells are successfully pillated in a small rotor at 10,000 rpm for 5 minutes. If we now use a bigger rotor with twice the diameter, so the diameter of the rotor is twice from the previous one, to plate the cells having the same fixed angle in 5 minutes. So the time is constant and the diameter was doubled for the second case. Now you have to determine the new RPM. So to solve this kind of questions, you just have to have a knowledge on a equation, that is the relation between the diameter, that means the radius, and the RPM. So the equation is like R1 into the RPM1 square, that should be equal to the R2 into RPM2 square. That is the for the first case, and that is for the next case. Just put the numbers here. 10,000, that is the RPM. So 10,000 square We don't know the radius, so we can take it as a r1, and here you can take it 2 of r1 because the diameter was twice. That means the radius was also twice. So now you have to determine this rpm. This thing you have to determine. Now it will, that r1 will be cancelled out. So now your rpm square would be like 10,000 square by 2. So your rpm should be 10,000 by root 2, which will come around this 7,000 rpm. Just you have to memorize this equation, and this kind of questions are repetitive in this paper. Just remember this equation and you can get the marks. So it is very easy. Here you can see certain enzymes and you have to determine which one adds extra adenine at the 3' end. So adenine is a DNTP. To add it in your target DNA or in a DNA sequence on the 3' end, there is an extra adenine. So it should happen via the polymerization activity. So here, cause only the polymerase can add the nucleotides. So here, except the topoisomerase, all are having the polymerization activity. The topoisomerase, they're having two types. One is the topoisomerase 1 and another one is the topoisomerase 2. The first one can introduce a single strand break and the second one can introduce a double strand break. And depending on this break, they can form and reform the coiling and supercoiling of the DNA. So it is very much important in the case of DNA replication or transcription. So the topoisomerase is the enzyme that is responsible for making the coiling of the DNA molecule. So it doesn't have any kind of polymerization activity. So the fourth option is wrong. If we look on the different kind of polymerization activity in the DNA polymerase, you will get three kind of properties. One is the common one, that is the 5' to 5' to 3' polymerization activity, that is related with the replication. The next one is the 5' to 3' exonuclease activity, and the third one is the 3' to 5' exonuclease activity. So this activity is related with the DNA repairing of the DNA, and this one is related with the proofreading. So how it works? Why there is a need of the proofreading activity? If via the DNA polymerase, this polymerization activity during the replication, if it has mistakenly introduced some wrong nucleotides, then the 3' to 5' exonuclease activity will be activated there, and it will remove the wrong nucleotide. That is the proofreading activity, and that will be repaired via the 5' to 3' exonuclease activity. And in the DNA polymerase, certain different domains are there to do different kind of activities. So here in the clean DNA polymerase, that is the DNA polymerase which doesn't have this activity. Why? This activity is not there. What is the activity of the repairing of the DNA polymerase? So that is deliberately not present in the clinal fragment because it was used in the case of DNA sequencing via Sanger's way. So if you remember the Sanger sequencing, there we are intentionally introducing a wrong nucleotide, that is DDNTP on the 3' end of the synthesizing DNA. So deliberately we are introducing a wrong nucleotide and thus the chain gets terminated. If this DNA is having the repairing activity, then what will happen? Then this thing will be removed and that will go on. So the chain termination is not possible if you are using the conventional DNA polymerase. So the Kleno DNA polymerase was designed in a certain way so the domain that is related to the exonuclease activity, it was excluded from the Kleno DNA polymerase. Now the DNA polymerase can only be used in the case of Sanger sequencing, where intentionally you want to introduce a wrong nucleotide. So without this activity, because if that activity was there, then the chain termination was not possible. So that is the utility of the Kleno DNA polymerase. And the next one is the reverse transcriptase. The reverse transcriptase is related to the production of DNA from the mRNA. So from the mRNA sequence, at first it will produce a hybrid, DNA RNA hybrid, and thereafter taking the RNA as a primer, it will form double stranded DNA. So it is also having the polymerization activity, but both that Kleno and reverse transcriptase are not able to introduce that 3' adenine residues. So the answer is the tag DNA polymerase. So tag DNA polymerase has an inherent property to introduce the adenine on the DNA that is getting synthesized. That is, if that is the 3' to 5', then the DNA replication will happen to the 5' to 3', and after that there will be extra adenine. So that is its inherent activity, and depending on that, if you want to use this activity when you want to clone your target, suppose your target DNA sequence via the PCR, you got that you have used the tag DNA polymerase, then on the 3' end you will get a extra adenine. And now if you want to clone this DNA fragment, so there is another DNA fragment. If you clone it with this, then you can make a 3' T. So your vector, if you are producing a vector that is having the T, the extra T on its 3' end, so that can come and attach with the A. So if you have designed a vector that is having a extra T on its 3' end, then it can form like this. So that is your vector and it is your target sequence. So here, so you can design a vector having a extra T at both the 3' ends that can come and attach with the A of your DNA that you have synthesized, that you have made via the PCR by using the tag DNA polymerase. So that kind of cloning is known as the TA cloning. So depending on this activity, you can make a TA cloning. So the right answer is the tag DNA polymerase.